So without further ado, let's kick off, and I will introduce you to Dr. Robin Sanderson, who is a consultant hematologist at King's College Hospital, NHS Foundation Trust, and we will get going from there. So Dr. Sanderson, over to you.
Thank you very much for asking me to speak. So obviously the topic has been explained. I'm a consultant hematologist at King's. I'm also in charge of apheresis, and I work with CAR T lymphoma patients. Next slide. So just a bit of background about King's. We are a very high delivery centre for chimeric antigen receptor T cells. So we were the first centre in the UK to deliver commercial Yescarta, which is one of the big products for high-grade lymphoma. So we have one of the largest CAR T practices in Europe. I won't talk anymore other than just to say this is on the background of a phase 1 allogeneic CAR T trial, and we also have multiple other CAR T studies, so particularly in myeloma and other types of lymphoid disorders. Next slide, please. You have to press it a few times so the whole slide's once more. Yeah, thank you. So this slide is really to summarize my practice. So I really focus on giving CAR T cells to patients with lymphoma. The two licensed products in this space are Yescarta and Kymriah, and more recently a third product, Tecartus. So these are all for different subtypes of lymphoma. And we predominantly deal with two companies, Kite Gilead and Novartis, and already the landscape is quite complicated. When we collect peripheral blood mononuclear cells to be made into CAR T cells, already the companies ask us to do different things, and what happens to the cells when we make them, different things happen. So already, even with just Kite, sometimes we send the cells to Amsterdam, sometimes we send the cells to Los Angeles. When we send cells for Kite manufacturing, we send fresh cells. When we deal with Novartis, we send cryopreserved cells. Those cells can go to the USA or Europe. And there's a new product coming, Lysocell, which will also go to the United States, and we will have to make cryopreserved cells. So already there's quite significant differences in what we're being asked to do, and that's just talking about the starting product and how to make these CAR T cells. Next slide, please. So this is just to summarize the treatment algorithm in lymphoma. So when we do CAR T, we would start with leukapheresis, so we collect the appropriate cells. Everything that follows from there is really timed in a way that we plan it at the very beginning. So patients will have leukapheresis for the lymphocytes, and then we will know how long the manufacturing is going to take, whether it's 3 or 4 weeks. So we during that time period, we will plan what treatment they will need. So some patients will need chemoradiotherapy, and after that, the admissions will be planned. As much as we can receive CAR T cells any working day, we can actually only start the chemotherapy prior to CAR T on Wednesdays to Fridays, because we can only infuse CAR T cells during the week. So there's already quite a lot of logistical steps that we need to think about at the very beginning. And I think you really have to remember the patients who are eligible for CAR T and lymphoma have chemotherapy refractory disease. If these patients get too sick, we do not infuse them. So delays in infusion, logistical problems really results in patients being palliated, because NHS England defines very specific criteria as to who we are allowed to infuse CAR T cells to. If they get too sick, we cannot proceed. We can't give CAR T cells to people who are literally dying. So it just really shows you how important the logistics are in what is a very finely tuned pathway. Next slide, please. We start with apheresis. Collection of cells is fairly standard procedure, but each company is asking quite different requirements from us in terms of labeling. So we have to have established SOPs to deal with the specific requirements from each separate company. Then as I've already said, some companies will ask for cryopreserved cells, some companies will ask for fresh cells. The advantage of being asked for cryopreserved cells is we can actually do the apheresis procedure whenever we want, to give us a lot of flexibility as to when we do it. That could also potentially mean that we collect cells from a patient who we think is very high risk, who we think could potentially need CAR T later. This is an area of active research in the CAR T field. We don't currently do it, just really because there's no funding for it, but it is sort of an interesting area in which sort of the logistics will alter our practice. If patients ask for fresh cells, that actually means that the day that we collect cells have to be coordinated with the CAR T manufacturer, because there's limited companies that are making these cells. That means we actually have global competition to make sure that our apheresis slots coincides with CAR T manufacturing slot at the company. So when we send fresh cells to one company, for example, they still need to be cryopreserved. So that actually introduces an extra step. So for one CAR T product with Kite, we just send the cells to Amsterdam and they get manufactured. For another CAR T product with Kite, they get sent to Amsterdam, cryopreserved there, and then sent to Los Angeles. So it really is quite complex. Each company, each product has different requirements. Next slide, please. In terms of manufacturing, just keep going. So let's just do a few, yeah. So one company with manufacturing will always offer us a consistent turnaround time. Another company that we deal with tends to estimate a delivery date, but then move it forward if the manufacturing process permits. This may not sound like a big difference to you, but to me, this is a hugely important difference. The company that tends to bring forward the manufacturing date, I must say, I don't really find that very helpful. What I really appreciate is a company offering a consistent turnaround time, because lots of things have to happen to the patient after we collect the cells before they can have the CAR T infusion. And I need to make sure that that patient has gone through all of those steps and is still in a reasonable clinical condition to receive the cells. So when the delivery time gets changed at the last minute, that really, doesn't help, because the patient still has to go through all these other steps under my care in the hospital. Also, between the two main CAR T providers, there are significant differences in manufacturing failures and out of spec products. Again, just to reiterate, a failed manufacturing product, a product that we don't receive on time, results in palliating a patient. Both of the companies we use, I believe it's a Salesforce-backed web portal. This is a novel thing for a doctor to be using these things. We don't commonly use these sort of websites when we prescribe patients chemotherapy. I personally find it very, very helpful because at least you get real-time information. One of the companies we deal with actually now allows viewing of available manufacturing slots. I think that's really very helpful because it helps with coordination of the whole pathway. Next slide, please. Just go on a few, just one more. I think just to stress, NHS capacity is always going to be a big factor for us. Specialist units are required to deliver CAR T. At the moment, there's 10 centres in the UK delivering CAR T for adult patients. Again, the logistics pathway does influence how we do things. So the cells will have QP release, so they'll pass all their quality tests in the manufacturing centre. And often, because patients are clinically progressing with their lymphoma, there is a rush to start them on chemotherapy to stabilise their condition. So there is a difference with the way companies approach this in terms of passing QP release and then how long afterwards the cells will actually arrive in my centre. So I wouldn't start the lymphodepleting chemotherapy for a patient until I knew the cells have actually passed QP release. But there then may be a gap between the actual QP release and the cells being received. And different companies do this in different ways. So that's, again, something to be considered, just the quality, how the quality control process happens at the company and how that's communicated to me as a user actually influences the management. Next slide, please. Okay, so, and then just to talk quite a bit about how we receive cells, the way we interact with companies is getting, as I've mentioned, increasingly complex. There's really a lack of standardization in the actual bags that companies produce. And a new company we're dealing with is actually proposing giving us vials, which would be something quite different. You really have to think very carefully about what sort of products are being produced. Another company is talking about doing dose adjustment. So they release like a finished product that then has to be modified when we receive it. So that means we have to take cells out. That may sound like a simple thing to do, but actually it's really not when you have to ensure the absolute sterility of these products. It's not practice with the commercial products for these bags of cells to be overwrapped, although overwrapping is something that is very helpful for us because it allows us to co-locate products. Co-location of products from different companies for us is a big issue. You know, we're a hospital in central London. We are landlocked. We don't have lots of additional space. So the way that cells are delivered to us, really, we have to think a lot about it because we have limited space. We have limited tanks. At the moment, we're keeping different companies' cells in different tanks. When we're dealing with new companies, it might be that they want to deliver a product in a dry shipper, but then that product would need to be delivered in a just-in-time way, which we can do, but it's just adding again to the logistical complexity of the process. It would be preferable that we have products that can all be co-located together, that we can receive a few days before the patient is due to be infused, so we know that the product has been received safely. But the increasing diversity of CAR T-cell products on the market, dealing with different companies, they're all obviously industry-backed, so they're different companies, is making our life much more complicated in this regard. Next slide, please. So really, I'm just going to conclude. You know, the autologous CAR T pathway is bespoke. It's highly complicated. We deal with patients who have refractory disease, so their window to get to CAR T infusion is very small. And if there are logistical delays, this will lead to patients not being able to have their CAR T infusion, and in some cases could actually result in palliative care for that patient. So now I can introduce my colleague, consultant hematologist, Dr. Ruben Benjamin. Thank you.
Yeah, so like Robin, I'm a hematologist based here at King's College Hospital, and I run a research group focused very much on allogeneic CAR T cells. And we've been very fortunate to work with the very first allogeneic CAR T cell trial globally. And the reason why we're interested in allogeneic CAR T cells is because whilst we've done a fantastic job with delivering autologous CARs to patients with cancer, there are some challenges associated with this. And Robin has already highlighted some of the possible challenges. Manufacturing failure, although overall it appears to be fairly low, it can't be discounted entirely. And there are a number of patients who are lymphopenic who actually don't get as far as having an autologous product being generated. The logistics of delivering autologous CARs is not trivial. And again, Robin's very nicely highlighted all the different challenges that we face with that. And probably one of the biggest challenges is patients who present with rapidly progressive disease. And it's asking an awful lot from an autologous CAR T product to actually overcome the disease of the patient who's declining in front of us. From patient to patient, the T-cell profile is incredibly different. And so you might expect the quality of the CAR T product to also be very different. And this ultimately impacts on efficacy. And cancer patients are well known to have dysfunctional T-cells. And I guess above all, the costs involved with delivering autologous CAR T cells makes this really not feasible for many countries globally. And so it's to try and address some of these challenges that allogeneic CAR T cells have had quite a lot of interest now in the last few years. And the basic premise behind this is that you have a treatment that's available off the shelf because it's pre-manufactured. These cells have already been validated and tested extensively, so we know exactly what we're going to be delivering to patients. You know, if you have a faulty batch made, you just discard it and generate a new batch, something which you cannot do with an autologous product. And ultimately, it offers the economies of scale where you can generate large numbers of CAR T cells from a single donor, for instance. And because these are CARs delivered, generated from normal, healthy individuals, it does have the potential to be more efficacious than cancer patient-derived CAR T cells. But there are two clear obstacles here. One is graft versus host disease, something that you see if you have CAR T cells that are not matched with the patient that you're infusing into. And the second problem is rejection of these CARs by the patient's own host immune system. And so the way in which the field has tried to overcome this is to essentially edit these cells, do further genetic modification of these CAR T cells to remove the T cell receptor that would otherwise cause graft versus host disease. But in order to try and prevent the patient from rejecting these mismatched cells, you also have to suppress the host immune system. And one of the agents used for that is an antibody called Campath, or anti-CD52 monoclonal antibody. But to make the infused cells resistant to the effects of Campath, you have to knock out CD52 in the CAR T cell. So this is just one of the strategies that's been developed in some of the early off-the-shelf CAR T cell products to be able to take cells from a single healthy donor and infuse them into multiple patients regardless of HLA type. And this was the product that went into clinical trials here at King's and other centers, CAR-19. And in this particular CAR T cell product, you have a CD19 CAR being expressed in the usual way. You have the TCR alpha gene knocked out using TALEN gene editing technology, and CD52 has also been knocked out using TALENs. And in theory now, this cell can be infused into any patient regardless of HLA type. And the results of the first trials were published back in towards the end of 2020. And in this study, which was looking at pediatric and young adult B-ALL, patients with relapsed refractory disease were offered a single infusion of this universal UCAR19 product. And the results showed that overall 67% of patients achieved a complete remission following treatment with this product, most of whom then subsequently went on to have a allogeneic stem cell transplant. If you were to look at a sub-cohort of this, so those patients that received a more intensive lymphodepletion with fludarabine, cyclophosphamide, and alemtuzumab, 82% of these patients achieved a complete remission. So I think this was fairly impressive results showing that allogeneic CAR T cells can be given to patients safely. You can get remissions. The problem came from the longer term outcomes, so that by 6 months, even though 90% of the patients who received intensive lymphodepletion responded, there was a significantly high relapse rate, so that by the time you got to 6 months, only 27% of patients remained in remission. This compares to over 50% of patients who receive the autologous CAR T cell product who remain in remission. So why is it that the responses were not durable? Well, we think that one of the main reasons was the lack of persistence of these allogeneic CAR T cells. Shown here is a graph that illustrates that CARs expand by the time you get to about 2 weeks, which is kind of what you see with the autologous product. But after about 2 weeks, the CARs decline in the peripheral blood of these patients, so that by the time you get to about 28 days, most of them no longer have any detectable CARs in the peripheral blood. There were a handful of patients that did have CARs that persisted for longer. So we think that it is the lack of persistence of these third-party CARs that results in the tumor, unfortunately, coming back. So how do we try and overcome this further? How do we improve the persistence of these allogeneic CARs? Well, again, the field is offering technological solutions to this. One of the solutions is to try and further engineer these allogeneic CARs. So we've already spoken about editing the TCR alpha gene and CD52 gene. If you edit out the class 1 MHC, so people have looked at editing beta-2 microglobulin. So now you have a cell that is more invisible to the host immune system, making it less likely to be rejected by the host cells. Another way of trying to further improve persistence is to introduce additional elements, HLA-G molecule, for instance, which has an inhibitory effect on NK cells, another host immune cell that can mediate rejection of these CARs. So whilst the very first study, the CALM and PAL trials were very encouraging in terms of early responses, perhaps rather disappointing in terms of lack of durability of responses. I think the field is now moving ahead, and I'm sure we will see further next generation CARs giving us better results in terms of allogeneic CARs. And people are not only looking at T cells as a source of off-the-shelf CARs, people are looking at putting in CARs into other cell types, so NK cells, iNKT cells, gamma delta cells, and then the holy grail to actually generate induced progenitor stem cell derived CAR T cells. All of this are in fairly late stage preclinical and clinical development. And I think you'll hear more of this in the next few years. So if I could just summarize that allogeneic CAR T cells currently can be an option in clinical trial settings where autologous CARs perhaps may not be able to be used. We have demonstrated the safety and feasibility profile of this, but we still do need to overcome issues relating to toxicity and lack of persistence. And some of this is being done through the development of next generation allogeneic CARs. But I want to now just switch slightly to another way in which the field is looking at improving access to CAR T cell therapies, beyond just allogeneic CARs. And one way of doing this is to introduce automated systems of CAR T manufacturing, which significantly reduce the time for generation of autologous CARs. So many of you will be familiar with the Miltenyi Clinimax Prodigy system, which can generate CARs within about 8 days and sometimes even less than that. There is another system that's now rapidly gaining in the field, and that's the Lonza Cocoon system. Again, very similar principle, but you have a fully automated system with conditions that are monitored very closely and where you can generate CARs within a matter of days. And some of you may have heard of even faster ways of generating CARs within just 24 to 48 hours. So I think the whole field of CAR T cells is now moving from the early generation of autologous CARs to allogeneic CARs and more automated, faster ways of generating CARs. So, I want to just kind of end by showing you what the UK kind of CAR T setup is like. I mean, Robin already mentioned that we have about 10 adult centers offering CAR T cells in England and another 2 in Scotland and Wales. Now, this may be fine to deliver CARs to just lymphoma and ALL, but as we start to get other indications being approved, for instance, multiple myeloma, which is now approved in the US and very shortly, hopefully in Europe, it's going to be incredibly difficult for just 10 centers to deliver the CARs in a timely fashion to patients that need it. And so undoubtedly, we're going to see a significant expansion in the number of CAR T centers in the UK and globally. And whilst I think the setup here in the UK and probably in much of Europe is actually not too bad, I think it's a sobering thought when you look at this particular slide. This is from 2 years ago, showing CAR T delivery globally. And you'll see that this is very much restricted to the North America, Europe, China, Australia, Japan, for instance. The entire globe otherwise is relatively poorly served when it comes to these cellular therapies. And I think dealing with some of these logistical challenges, trying to develop new products that can be cost effective and delivered quickly is really the solution to try and improve access to these therapies globally. And I want to sort of end here basically by a quick thank you to kind of all my colleagues who work with me at King's College London, as well as the clinical teams at King's College Hospital and Guy's Hospital, in order to be able to deliver some of these novel therapies to patients. Thank you. So I want to now hand over the stage to Mark Buch from Vineti. He's a Senior Vice President, and he'll be telling you more about some of the software solutions, I think, for cell therapies. Mark.
Great, thank you, Dr. Benjamin. And I'm going to be echoing, I think, a number of themes, maybe from a slightly different perspective that were covered by both Dr. Benjamin and Dr. Sanderson. So I appreciate being here today, and thank you, MedCity, and also the London Advanced Therapies Network for inviting me to participate in the talk today. Hopefully you'll find it useful. So just a little bit about Vineti. My name is Mark Puich. I oversee the business development and sales for our company, and I'll kind of dive into a little bit in a few slides, kind of what we're doing within this space. So if you could go to the first slide. So I think what is being highlighted here is this massive complexity that is coming with these new advanced therapies. And our chief commercial officer comes out of Dendreon and sort of recognized some of the challenges of these products going way back, decades prior. And what he calls it is a 3D game of chess. You've got what is the patient journey, you've got the product journey, and you've also got in the commercial products this financial journey of how all of these steps that are existing within a treatment process need to be executed in sequence, reliably, at scale, and with very low risk to patients, really ultimately is the challenge that comes with these therapies. And so when we look at this kind of value chain, this is focused on the clinical context, you can see how many different steps stakeholders are involved with, especially in autologous therapeutic. And tying this all together is really the challenge that Vineti is trying to play in this space. So if you go to the next slide. So our company was founded in 2016 with a leadership team that really came out of the cell therapy world. So one of our co-founders, Heidi Hagen, was the SVP of operations at Dendreon for 10 years as they launched Provenge, really set the stage from a regulatory operational standpoint on how the industry was going to view a personalized therapeutic, what needed to be managed in terms of chain of custody, chain of identity, and managing the massive logistical complexity of quite a high scale therapeutic. I think they're still doing on the order of 10,000 doses a year, which still dwarfs the current CAR Ts in terms of volume. And so that was really the inspiration for Vineti. We were founded to solve the problems, unique problems that were coming with these therapeutic CAR Ts that were moving very quickly through the clinic. And Kite became our lead customer to help launch Yescarta initially in the US in 2017. So we've been working with them, supporting thousands of patients, hundreds of clinical sites as they've rolled out their product to more and more organizations. And in the meantime, we're now working with about a dozen other customers, a mix of commercial or commercializing organizations. So we have another CAR T therapy that's in Europe, or excuse me, another autologous therapy that's in Europe, and then a number of clinical stage companies and a mix of allogeneic and autologous therapeutics that are all working to scale up their studies and ultimately support more and more patients over time. And at our core, we are a technology company. We have a software product that we've developed that is configurable to manage the unique complexity of these therapeutics and really tie together those key stakeholders. And you'll see that a little bit later in the presentation. Just to kind of highlight a little bit more about kind of our organization, we're now 180 employees strong and continue to grow to support a global market that we are seeing in this space. And I think we are ultimately looking at this built for purpose platform as being really the only way these therapies can be supported at scale. And I'll talk about some of the challenges and echo what was highlighted in the prior conversations. So if we go to the next slide. I think what was really apparent from both our co-founders' experience in this space and then most recently as we continue to work with more and more organizations supporting this industry is the feedback from the clinical sites is that there is frustration, there is confusion, there is concern that as more and more of these advanced therapies come into their centers, all with different expectations from each manufacturer, is going to become very difficult to manage. And so this is really something that we have been focused on from the beginning is how do we streamline and make the practice of medicine around these therapies as simple and easy as possible for really the key partners in the clinical sites to be able to support more and more patients at scale. And so this has been something that I think Dr. Sanderson highlighted of kind of all the different apheresis processes that are coming through. And really at the end of the day, there is a very unique relationship for these therapies between the healthcare providers and the pharma companies, one that is more intimate and different than has really occurred in the past, especially at the scale that these therapies can be rolling out to. So that is kind of one of the key challenges that we're seeing in this space. If you go to the next slide. I think another one is sort of the massive kind of complexity that is growing as companies roll out to more and more studies and more sites. So when you look at kind of that initial, kind of before the slide was building, that initial step, an initial study in a single region with a single manufacturing site, kind of relatively easy to get your head around. You're maybe dealing with 5 to 10 clinical sites. You can kind of standardize that study. You have the CROs that are in helping you. But as you start rolling into more and more sites, more and more regions, and if you continue to build the slide, you see that the complexity just becomes massive for the manufacturers. They have to deal with hundreds and hundreds of clinical sites and hospitals, each with their own procedures and processes, now needing to be able to provide and feed data back and forth between the manufacturer in different languages that are local and tailored to the local region, and also dealing with different regulatory expectations within different countries. So France may have slightly different labeling, handling of patient data that may be different than Germany, than UK, than the US. And so dealing with building out a supply chain that is going to be able to address all of this complexity around the world in a localized way and very quickly, because we do see these therapies rolling out globally as quickly as possible to maximize patient access, becomes an extremely difficult challenge for the manufacturers. And so a software product like ours is really trying to allow this sort of expansion to happen in a way that's not going to become crippling from an operational standpoint for the manufacturer, as well as difficult for those sites that want to actually treat these patients. And we're also seeing a number of manufacturers explore therapeutic options beyond the autologous. So they may be starting in autologous, but obviously have active allogeneic programs for all the reasons that Dr. Benjamin highlighted. And so running all of these different programs with different flavors of allo, some of which are matched, a little bit more closely to patients, becomes, again, a huge logistical challenge that is ahead of all of these manufacturers. Next slide. And then I think we can't ignore the access question that was highlighted in the prior presentation as well. Most patients are not near centers of excellence. So as more of these therapies become more safe, as they become something that can be treated more in a community setting rather than a COE, how you now manage even in an increasing complexity, how are you getting it to more local and distributed environments that aren't close to cold chain centers that can have overnight delivery. Those are some of the technical and operational challenges that are going to be facing the industry to really reach a broader number of patients, especially as you start thinking about some of the rare disease indications where maximizing that patient population is going to take reaching a broader market more effectively. So next slide. So one solution and one really core fundamental principle we believe in is that standards will be key in sort of driving this industry forward. That if every company, every manufacturer is bringing their own processes, their own tools, their own systems, their own procedures into every hospital around the world, that will be unsustainable. And so this idea of industrializing the market becomes really a key principle that I think us and our customers are targeting. And so Vineti is providing a solution that is working across a number of manufacturers in the space, where our goal here is to really move from left to right, which is tying together all those stakeholders in a very automated way, where we can, building automated integrations of our software into key stakeholders like Lonza as a CDMO we announced as one partner, so that as more and more companies come into the space that are running studies and launching products, they don't have to rebuild the supply chain from scratch every time. They're really kind of leveraging the rails that have been laid by a Vineti system into hospitals and across the supply chain. And so they can focus on the science and the patient treatment and all of the services they want to provide around maximizing the success of their therapies in those clinical sites. Next slide. I think standards also plays a role in other ways. We are an active member of the Alliance for Regenerative Medicine and the standards coordinating body, which is one group that is trying to drive standardization of across a number of aspects of cell therapies, from assay development to logistics to assay approaches, or excuse me, apheresis approaches. And we have been active in looking at this idea of the chain of identity as one area where right now there is no standard around how a patient is identified, how they remain unique across all of these stakeholders in the supply chain. We've come up based on our experience looking at personalized cancer vaccines, allogeneic therapies, autologous therapies of different flavors, solid tissue-based, CAR-T-based. You start seeing there can be quite a lot of complexity in the ways tissues are collected, where they're collected, how they flow between multiple different supply chain steps, and finding a way to come up with a standardized way that all of us can uniquely identify these patients across these different processes is going to be I think, fairly critical to assuring patient safety and a more streamlined approach to bringing these therapies to more patients overall. So I think that's all I had. So I want to appreciate, again, the opportunity to speak here, and happy to take questions when we get into the panel part of the day. I'm going to be passing off now to the next speaker. It's Thomas Ellman, who works for CSI Life Sciences and Healthcare, logistics company within the DHL world. And take it away, Thomas.
Thank you, Mark, for the intro, and welcome everyone, and thank you, London Advanced Therapies, for giving me a chance to present here. So next page, please. A little bit to my background. So I have a scientific background as well, but actually have worked all of my professional life in supply chain and logistics function, of which the second half of it with DHL in life science logistics. I'm serving as a member of our senior management team, and I'm looking globally after our clinical trial logistics, and that includes also upcoming solutions for cell and gene therapies. And I'm also dealing with the vaccines distribution, where DHL is quite active in the last couple of months. Next page, please. Some of you may think DHL, isn't that the company with the yellow vans and the guy who clears the parcels in the room behind reception? Yes, we do that as well, but actually I'm one of 9,000 associates who are dedicated in the life science space. We have also 250 pharmacists in our warehouse landscape around the world, 170 GMP qualified spaces. We have a clinical trial depot network, and obviously we have large networks, be it air freight, express, or ocean freight, that handle the cargo. And we obviously put huge efforts in terms of training, qualification of our associates, certification as well. We're also a member of GS1, as Mark mentioned, standards and defining standards on the way forward. That infrastructure, the next slide, please, also helped us to basically now ship almost 500 million doses of vaccines in the last 6 months for all main manufacturers. Obviously, vaccine distribution is not the topic of today, but what I want to stress here is that the rapid development not only of the COVID vaccine, but also the rapid development around the logistics around this have helped us a lot, and we see also impact now going forward into personalized medicines, personalized treatments, and cell and gene treatment. One of the things, for instance, that we've developed is real-time tracking of the vaccines, and most of the vaccines that we're shipping, especially on the RNA base, are going with our express network, and that has had huge benefits, especially in the earlier phases of the pandemic, when huge parts of normal commercial uplift was grounded. And when you think about how you treat your dry shipper and how you send your dry shipper for the treatment around the world, if commercial airlines are grounded and you basically have no other solution, our fleet was up in the air and basically helped to ship these vaccines around. So now talking about trends, and that's on the next page. What we see from the logistics side is very much that customers, and our customers are big pharma, but also startup companies coming to us, and they're concerned about these 4 pillars. So today I'm not talking about sustainability, but that's obviously on everyone's mind as well, or digitalization, which we see as a huge trend. But here on the 2 left pillars, everything around advanced treatments, and be it now developments on RNA technology or cell and gene treatment, and what can we do in order to make our customers more successful. And what we see a lot is that this whole industry becomes, someone said that we become smaller and colder. So we see less volume to be shipped, but higher values, very delicate treatments, and obviously covering different temperature ranges from our normal business of controlled room temperature of 2 to 8 degrees, now going into -20, -70, or liquid nitrogen treatment. So on the next page, this is a typical lead of what we would got from a global biopharma company. They're obviously looking into the future and see how they can make the movement of cell and gene therapy treatment and personalized medicine more economic. It is covering those temperature ranges that I just mentioned, from -20 to -150 down to -196 degrees Celsius. And it's really basically the distribution around the world. They are concerned about not having their product hold back in customs, obviously, and eventually running out of temperature. And it requires basically all of us with the different divisions that we have within DHL to come up with cross-BU solutions and go into specific investments. And some of them you see on the next page. And if you would click further. So obviously some of the things that we're doing here is, and that is the previous page was basically the bird's eye view of one of our flagship biotech campuses in the Netherlands, Nijmegen. From the outside, it looks like a warehouse. This is actually the place where we have installed, for instance, something like 150 ultra-low freezers, you see on the next page, to store vaccines and to distribute from there, right? Those vaccines that we basically ship around the world. But we have other areas there as well where we're going into huge investments. So you go one click further. So we have GMP areas as well. And we equip them with the capability to handle also what is required in terms of the temperature ranges for cell and gene therapy. So the next page shows you some, obviously, of the things we use for transport, but also for long-term storage. As Professor Benjamin was talking about the research on allogeneic, that is obviously the one thing we're very much looking forward to have as well, because we believe as well from a logistics point of view, that is where the whole thing becomes much more standardized, easier to handle, and also easier to scale it up and to bring it in a very cost-efficient way. So talking about these processes, on the next page, I think we have seen that is interesting what happened to my colors here on my presentation I sent to you. Basically, the autologous process, which is very complex, circular process as well, with a lot of moving parts and a lot of things you need to manage here on the different flows from the patient into the laboratory and back to the patient, also with the provision of the temperature provisioned dry shipper already. So very complex to measure. And versus that one, the allogeneic on the next one, much more straightforward. Obviously, it's much more similar also to what we see today from a finished product point of view, delivery from stock from a DC basically to all over the world. And we see that out of the site in Nijmegen, for instance, where we have orphan drug manufacturers, where we perform things like late stage postponement and ship it from there as a multi-market storage hub and as a DC all over the world. So much easier for us to handle. So obviously we are very keen to see the first allogeneic projects getting approval and coming to the market in a larger scale. Now, to basically go further on the next page with case studies and with solutions that are expressed to us. So here is a company that is in CAR-T therapy actively. And they ask us, what can you do as DHL to basically support our autologous flow? So what we see is that in this autologous flows, there are obviously different lags that have different requirements. What we do there when the product is really loaded with the cells from the patient is that we're using dedicated solutions with our DHL same-day solution. But where you have return flows of the dry shipper, that is obviously something that we can do much more effective and using our express network there as well. Obviously, we have some sort of control tower over the whole thing, and I believe also solutions like Mark mentioned that in his previous presentation will help us to create better standardization, because on the next page, you see basically what we have put in place here. Pickup of the dry shipper, bring it basically to the investigator site when it's loaded, goes to the customer site, and then back there. And on the lower swim lane, you basically see the flow back from the customer to the investigator site. So that is all done within dry shippers at liquid nitrogen temperature with different countries involved. This example here is basically based on German and Netherlands thing, which obviously is done by truck, which is for us the quickest and most safe solution. But wherever then we see more hospitals rolled up, and the next phase of rollout will be in the US, then obviously air freight comes into the game, and that will be largely then built on our express network. And then being conscious of time, the very last case study I brought to you is very similar to here. This already includes air freight because it is a switch between Belgium, Germany, and also Israel, where we perform an autologous flow where we have very close contact to the customer with our overarching control tower, and basically then observing the whole process. And every 15 minutes, 30 minutes, we give an update to the customer back on where is the status of the shipment and what is happening with the cargo there. That is in place since a couple of months now, works very seamlessly, and obviously just two examples of the things that we're doing here. And with that, and looking at the time, I'd like to thank you again, and I bring it back to Rick.
Thank you very much, Thomas. And thank you for everyone that's spoken prior to Thomas as well as the questions that have been coming through. So now we will go to Professor Francesco Dazzi, who will be moderating the panel discussion. And Professor Dazzi, if you could please, as well as all the speakers, if you could please put on your video and unmute yourselves, and we will kick off from there. Professor Dazzi, if you could please confirm that you can see the questions that have been coming through.
I can have seen everything. So there have been quite a lot of questions. I think first, congratulations, beautiful meeting. I think it's a really polyphonic choir, addressing all the different components. Can I just start? So let me start in sort of two blocks. At first, I tried to put one against the other, Ruben and Robin. There is a lot of, there were a few questions about standardization. And I think this is a critical question, considering that there are different efforts which go to a certain extent one against the other, which is, should we do a personalized medicine or an off-the-shelf? This has implication on standard, on efficacy, on a lot. Can I ask, comment from both of you about this? And then we come, of course, to the logistics, another layer of complexity. Ruben or Robin, do you want to start?
Yeah, so, my view is that, at present, clearly personalized autologous CARs have shown the results that have led to their approvals. And I think that is the gold standard now to work from. So for a allogeneic CAR T-cell to really get into the market, it really has to demonstrate that it is at least as effective as the autologous therapies before all the other advantages come into play. And thus far, unfortunately, that hasn't been shown to be the case. Whilst I've shown you some very encouraging early results, and I think it's fantastic for the field that those trials are now completed, but we still need to get better results to really say that an allogeneic off-the-shelf CAR would eventually get into the clinic. I'm sure it will come in, but how soon, that I can't say, Perhaps Robin can talk about autologous CARs in terms of the variability that you're seeing from patient to patient now, in terms of the specifications that you see, because you're involved with looking at the outer products that are out of spec.
Products, yeah. So most products are in spec, I suppose, to be fair to the companies, but there is this significant minority of patients who either have an out-of-spec product or have very rapidly progressing disease. But both of these groups would very much benefit from the presence of an allogeneic option. So I don't think it's really an either or for auto and allo. I think there's definitely space, certainly in my practice, for both types of product. But the out-of-spec I mean, the out-of-spec issue is I think just represents how complex the autologous bespoke pathway is. You know, when it works, that's great. But there's so many ways that a product can be out of spec. I mean, if you look at the QP release that the companies produce, I mean, there's many different tests that they do, and it's really quite complicated as well. I mean, there's things that are clearly out of spec, like I don't know, like bacterial contamination, but there's many things that wouldn't necessarily be clear whether they're out of spec or whether it would be clinically significant or not. So I suppose it's just sort of saying that the autologous pathway is working, it has very good clinical results, but there's plenty of scope to improve it. But I think the patients that fall off the autologous pathway, even within the licensed indications, there's definitely scope for allogeneic backup. So I would love to be able to do that.
Yeah, the question.
I have a comment just to kind of highlight. I think it's also not a binary in that there are a number of indications that with neoantigen-based solid tumor kind of scenarios with mutations that just won't be able to be managed potentially with allo, right, to deal with each patient's unique tumor profile. And so I think for some therapies like CAR Ts on the market, they can be more standardized. Others may need to be bespoke just by the nature of the treatment.
Although the bespoke does require a very solid assessment of the variability, because in particular with, of course, CAR T cell, the patient that are already third, fourth line of treatment, so that does clearly create a massive variability in terms of donor type. You know, Robert, what is your experience? I mean, there was a question even about vials versus bags. You know, that is banal to a certain extent, but it's big. Also, is cryopreservation now a GMP procedure at this point? Because, you know, it's fundamental. If you send the wrong cells, which is not being validated with a lot of process to the company, you may expect a disaster. Well, disaster, not a good product.
I mean, you're right about, I mean, heavily pretreated patients is definitely an issue, I think, in terms of the quality of the cells. I think particular issue of my practice when patients have bone marrow disease, that definitely seems to me resulted in a higher chance of a manufacturing failure. I mentioned quite a bit in my talk about the difference between companies requesting cryopreserved versus fresh cells. Clinically, that's really, really important in terms of the way we practice. So, yeah, I mean, again, allogeneic has all these sort of hypothetical advantages of having much healthier T cells that haven't been exposed to loads of chemotherapy. But then we still have again the bespoke autologous pathway is working for a lot of patients, but there's just still plenty of scope to improve it.
And Ruben, the question is also, so for allogenic cells, can you ensure the quality of the cells across the board? Is it very easy peasy, or is not?
I mean, I think the theory behind being able to take an apheresis product from a healthy donor and manufacture multiple doses of cells, to be honest with you, I don't think we're there yet. So I can speak from our own experience with the CALM trial, where we treated 35 patients in total. Sorry, 25 patients in total, and we had to use 3 different donors, and we had to use 9 different batches of cells. And the reasons for that was because in some cases, patients had anti-HLA antibodies, so we had to switch the donor. But in many other cases, there were batch failures, didn't quite meet to the specification during manufacturing, and therefore they had to generate a new batch. And genome editing of cells is not a trivial matter. What was used for the UCAR19 trials were talons that were electroporated into the cells, in addition to viral transduction of the CAR. But that process in itself results in significant cell death within the first 24 to 48 hours before the cells then recover. So it isn't the case that you can reliably say you take a single bag of apheresis product and generate this many doses consistently. I think that, to be fair to the manufacturers, they are improving things as we go along. And now there are more and more manufacturers in the allo space the gene editing technology is improving, the conditions are being optimized. So I think looking ahead, I think it could be far more standardization of the allo CAR product. The other problem we also have is trying to work out what are the best assays to use to define how functional the product is. We're still left with fairly crude viability assays, cytokine secretion assays, and potency assays that are not very reproducible often. So it's not entirely possible to take a given CAR-T product and say, look, we can guarantee that there would be this much efficacy in a patient. So I think the field is moving forward, to be fair, but there is still significant issues with allogeneic CAR-T manufacturing itself.
And just to go to sort of clinical practice, there was a question about, why did cells disappear in a couple of weeks? And if I may also add another question to this, what about, rather than, of course, very elegant, sophisticated further gene editing, to do 2 or 3 doses from different donors? Would an allogenic strategy, would make it possible?
Yeah, so the reason why the CARs don't persist for very long is because you get some degree of host immune recovery and therefore rejection of the CARs. This is despite using fairly intensive lymphodepletion, fludarabine, cyclophosphamide, and alemtuzumab. Despite that, you do get some recovery, whether that's T cells or NK cells, and that's causing the rejection of these products. And you don't obviously see that with autologous products. Now, can you actually improve on that? Well, I highlighted 1 or 2 strategies in terms of engineering of the cells that might result in a product that persists for longer. But multiple dosing, in theory, that is a solution. And in fact, if you read the Lancet paper, we described 3 patients where we did exactly that. We dosed them on 2 separate occasions, and in 2 out of the 3 cases, there was response a second time round. But you still get some host immunity, anti-murine element of the CARs that basically means that further doses from the same donor may not work, and you may have to switch the donor. So we're still learning the nuances of what second, when is the best timing for a second infusion? Do we need to switch the donor second time round? Do we need to lymphodeplete them second time round, or change the lymphodepletion second time round? There are many variables here, unfortunately, but we just have to work through some of these in the next few trials.
But basically, there seems to be a critical need of still controlling long-term the tumor. So 1 shot is not sufficient. So in this perspective, would allo or autologous, because they still got pro and cons on both sides. Can you just very briefly, before we pass to another side, comment on this?
Yeah,
because,
I mean,
one could be tolerance, and the other one could be just simply is not effective.
Yeah, so I mean, in terms of what causes tumor relapse in some post-CAR-T, one is antigen escape, and so second time round, there's no point in giving a second infusion of the same type of CAR. But in the majority of cases of lymphoma, for instance, the relapses are not due to antigen escape. So it must be something else. It must be either the CARs becoming exhausted because of constant exposure by tumor, and therefore they're no longer active. There's something about the immunosuppressive tumor microenvironment that inactivates the CARs. So that's where the field is looking at by adding in checkpoint inhibitors to try and see if that can overcome this resistance to CARs. So I think identifying all the different mechanisms of resistance to CARs is critical to decide how we improve the efficacy post-CAR-T therapy.
Robin, any further comment?
Just Ruben's point about why do CAR-T cells fail. It's just interesting when you look at why CAR-T cells fail in ALL versus lymphoma. It's different. In ALL, you often get CD19 negative relapses, and in lymphoma, you often get CD19 positive relapses. And I think they imply the 2 mechanisms that Ruben alluded to. CD19 negative probably as a result more of the tumor mutating and the effect on the CAR-T cells, where a CD19 positive progression is more likely to be, the tumor microenvironment sort of blocking the CAR-T cells getting to the tumor. So there's so much work to be done, and I think that giving CAR T cells with something else is something that's interesting to me. So not with chemo, not with cytotoxic drugs, but with other antibodies or checkpoint inhibitors, or other things that can manipulate the microenvironment with the CAR T cells.
I wonder whether Mark has anything to add, considering the multi background that Vineti has in terms of what you learned with antigen-specific T cells, basically.
Yeah, I'm not the scientist in the room, but I could speak. There was a question you had embedded in your first question about the cryopreservation, and one of the challenges for manufacturers is how you deal with this complex network with fresh versus cryopreserved cells to really be able to scale patient access. What the feedback we've heard is cryopreservation reduces some of the effectiveness of the cells, and then also how do you keep it consistent? So if we do want, say, an apheresis product to be cryopreserved before it ships to manufacturing, maybe we're sending it across the ocean for processing. What we've seen is companies looking at third parties where they'll send fresh cells to one location that can do a consistent cryopreservation process across a whole region before the shipment, to assure at least some consistency in the way it's handled. If you go to every hospital, they may all handle it slightly differently. Same as on the thawing process. How do you assure some consistency in the way that you're kind of rejuvenating the cells for treatment? So that is something that I think our customers are really exploring, different options about how to handle this. And with some preferring to keep fresh shipped, but then there's significant logistical challenges with everyone being aligned on the patient needs to be ready that day it ships, or else they can only be held for a day or so. So that was more to one of your earlier questions.
I would like now actually to put Thomas and you, Mark, on the spot. I think that cell therapy distribution is clearly a great example of a super complex logistic we've probably never come across, apart from the pandemic, of course. I just would like to both of you to comment and elaborate on again a sort of polarizing concept, which is the point of contact approach, whereby you can build your cell therapy at the bedside in a way or another, and the other is actually this fantastic infrastructure DHL has, for example, created by shipping all over the places. And in this, I wonder whether you can also elaborate in accessibility, low middle income countries, and what have we learned from vaccine? I know it has nothing to do now with vaccine, but that has been a fantastic, I think, experience to ship something that was usually shipped in a fridge at -80. So can you just both maybe starting with Thomas, who hasn't yet?
Well, yeah, thank you. And I think there were other questions as well in the chat concerning some of the customs and temperature monitoring and things like this. So obviously, I have to say, I mean, we're shipping since maybe 20 years life science products around the world, but that is coming normally in the range of controlled room temperature and 2 to 8. And I believe in the last 1 or 2 years, we really see that the trend goes to deep frozen temperatures. And we see that as an increasing trend. And I think with COVID, that will be even accelerated. So how do we do this? Obviously, we work together closely with partners, and partners can be airlines, can be ground handlers, but also packaging companies and temperature logger companies. And I think there are various solutions now in place in terms of active cooling or passive cooling. I think, and that was one of the questions here in the chat as well, the supervision is making big steps forward to online supervision versus passive loggers that you can only read out at the destination. There are a couple of loggers now available that are allowed to be on while in the air, and we see more and more progress there. So we use that intensively in the distribution of the vaccine, and I see that also swapping over to clinical trials, to cell and gene therapy, and so forth. But obviously, the temperature control comes with the packaging. And then within the air freight transport, obviously that is in a controlled environment and that it's in a safe mode. But when you're alluding now to low and middle income countries where we see the challenge with the vaccine distribution and also with cell and gene therapies is obviously the infrastructure in the country and the domestic distribution in the country. So there is, but that's another topic. We have intensive discussions with Ministry of Health governments of those countries and with NGOs as well, what to do in the country with the distribution. I think in the international distribution, that is all okay. And in terms of transport, and that was another question, we use actually both. We use commercial air freight with the airlines we work with. We have a next flight out service, but where we see that that was maybe not available because air freight have grounded their capacity, we use our express fleet, which is like 300 planes, and that was up in the air, right? And I think in the combination of that, we basically can cover 220 countries on the planet, but domestic distribution in certain areas is really a challenge. And that would be for all of these flows, yeah? So that's a little bit from my side.
Can drone help? Drones? I know there are studies in, at least
that's interesting as well. I think I do not see massive drones flying over central London for security points of view. Can drones go to remote places in Africa? Yeah, maybe. I think there was now in the last couple of years huge technology development in terms of the reach of these drones. When you see articles, this is going more now into hundreds of kilometers instead of maybe a dozen kilometers. It could probably be a tool to reach remote areas, but it will not exclude or change basically on the main legs, obviously the normal ways we transport goods as well. Mark?
Yeah, I think to the point of sort of the bedside manufacturing to central, I think we've seen with some of the recent BLA filings or attempted BLA filings, real challenges on manufacturing that still need to be worked out, even when you have full control over the environment for these therapies. So I think it's quite a ways off to have a process that's going to be so well understood, especially with variable incoming raw materials from each patient, that you could have sort of a machine in a box or a plant in a box that could put these out in a reliable way. I think what I'm expecting is more of a regionalization of the manufacturing that's already happening, but maybe at an increasing scale. We have partners, Lonza and Ori Biotech is another UK company that is a partner of ours, that are really pushing on these, kind of turnkey manufacturing systems that maybe could end up in hospitals, more likely in the midterm that would be still manufacturer run or CDMO run, but put more regionally to allow quicker turnaround time and local access to the therapies, especially for the personalized, for the autologous. In the allo case, we know that there's very, very big concerns within each hospital about storage of cryopreserved materials and keeping allo materials in inventory. There just isn't a lot of space. You hear of cryo containers lining up in hallways because there's nowhere else to put them. So again, even in the allo use case, we'll expect, I think, local or regional, cooling facilities like DHL provides or others to be able to do a more rapid distribution of these allo products when needed to community hospitals or centers of excellence. And there's also some interesting technologies. There's a company that has a cell therapy for stroke, a stem cell therapy for stroke, that requires treatment within hours, where maybe that model wouldn't even work to have regional storage. And they're looking at a vending machine approach within centers of excellence or emergency rooms, where they would store the cryopreserved vials, they would enter in a code, it would release the vial, they could treat the patient and also communicate back on inventory needs to restock this material. So there might be some creative things that happen if there really truly is a very short time to treatment that needs to happen with some of these therapies.
So there was a very interesting question from Katerina here in MedCity about the modular logistics. I mean, this is really a big question for both of you, because that could actually make a big difference. I mean, at the moment, we're still spending a significant amount of money just to distribute the product. So why is working on reducing the cost of the product manufacturing, there is also a need to reduce the cost of distribution, which isn't trivial. I mean, how can we address this? Because it's really is fundamental. Are they modular? Are you well, you both are somehow testing in different ways, this modular architecture.
But maybe if I can go first, I think for us, the difference is an autologous flow. Every flow is different and needs to be set up individually. And obviously need to be set up with an SOP and with a protocol, with testing, with validation, and so. Where we compare that then for allogeneic, that would be for us much easier as shipping out of a central depot, right? Where we then have the right tools to have a cryobank where the allogeneic cells are stored, and then it would be much easier for us because it would be much more similar to distribution of other now also orphan drugs that we do from central place. And it would basically require not so many locations on the planet to basically distribute it to a number of countries, because with our networks, we can be within 24, 48 hours, basically be on any, let's say, main airport here on the planet. That would be much easier and much easier to scale it up, versus autologous really needs to be designed and configured for each and every new setup on the involved laboratories, manufacturing sites, hospitals as well. Yeah.
There is clearly I don't know whether actually DHL has implements. Here there is also an issue with chain of identity, all the different components. So if we think about both allo or auto, type of product, there will be exactly as you say, the storage of the raw material, then of course the modification, then go back to another storage facility. How can this, and this implies, of course, quality assurance, it implies identity, resource, tracking back to every single component. It's not trivial, is it?
No, not at all. And I think as we think about, ways to drive costs out of the process for these therapies, the shipping is obviously a key piece. When you're not going, kind of international, that can reduce the costs. I think as volumes increase, so out of one manufacturer, maybe providing multiple doses to one site, they could ship together. But then this tracking and chain of identity becomes more complex. So I think a system like ours brings to the table sort of the barcode scanning and assurance that what went into a container is the same thing coming out at the other end, to really tie it to a patient that is getting treated. As you start looking at building sort of more efficiencies into the process, and maybe doing kind of combined shipments or multiple batches manufactured within the same facility, that tracking becomes even more critical and under more scrutiny by regulators to make sure that there's no mix-ups, right? Because that's the big concern we all have, is that something like that will happen and could set the industry back. And so it's non-trivial, and at scale needs software to really assure that those handoffs are appropriate, and so you can start building more efficiencies in and not manage it on paper.
Yeah, I think that standardization is really key to be able to scale it up and to be able to ship huge numbers. And also to drive the cost down, obviously, for everyone involved in the game, and to avoid errors. Yeah.
So that also means to have a large number of procedures to scale the price down, right?
To some extent. I mean, it's still a batch the size of one. I think as in the current world, as you build out more volumes, you're just putting more biosafety cabinets in your plant and more cryo containers moving around. But that will drive some efficiencies, and then again, these technologies for the automation side could drive some efficiencies as well. But you'll never get away from the fact that you're still doing one run for one patient in the autologous setting. So that's,
But still, even in the allo, there would be a donor pool. So again, there's going to be some issues with, of course, some significant. Can't say personalized, but it's not an antibody. That's what I'm saying. It's a much more complex. From the clinical perspective, I don't know whether Ruben and Robin can see this as a real option. I know you're in the end the operator, injecting, treating patients, but it's still something needs to be considered in terms of future investments. So again, the question is allo auto.
Yeah, I mean, one of the things that, just to follow on the issue about standardization, I think that is crucial. Even if you look at specific diseases now, like myeloma, for instance, where you have multiple opportunities to collect cells in order to generate a CAR-T product, the trials ongoing are currently being done in late-stage disease and now are moving upwards to second-line, first-line treatment. So I think that the nature of the product that you generate from a patient early on in the disease is likely to be significantly different to a product that's generated later in the stage of the disease. So I think it is very important that as manufacturers, that, well, the clinicians actually put pressure on manufacturers to find standardized ways of collecting starting material, freezing them down, even if the final manufacturing itself might be different. But I think the starting process, does need to be standardized.
Yeah, I mean, the autologous logistics do sound a bit more complex, just because you have to have, obviously, that goes from one person, has to come back to one person. Yeah, I mean, we've discussed the complexities of the journeys of these cells. It's certainly interesting as a clinician worrying about logistics, dealing with companies, web portals. It's very different to other parts of my clinical practice.
Can I ask probably the last question to both Mark and Thomas? Clearly, clearly increasing access, and again, go back to low middle income countries, would produce massive income for industry. So it is not something that probably industry would neglect. So on your perspective, do you think, because that is where you actually can be the center stage, how can you provide? Will you be able, I mean, is there any thought about expanding to low middle income country in whatever form or shape that is, of course, affordable?
I can speak for us. I think where we go is driven by our manufacturing customers. So we're not moving ahead of them and knowing where we need to be and what languages we need to support and sort of regulatory IT environment we need to exist in. So we know we're now being asked to move into Brazil and Argentina for some studies or even product launches next year. So we're sort of following the flow of our customers and where they see their markets evolving. But I think, Thomas, you're probably everywhere already for your world. Yeah.
I would say we need to distinguish in the DHL world between transportation and warehousing. Transportation, with our networks, we're covering basically 220 countries in the world. When it comes to warehousing, obviously that is in combination with our large customers, right? Where someone wants us to invest or co-invest or come up with maybe a multi-user facility, that is where we go. I mean, right now we're already at key markets. If we go into another, let's say maybe BRIC country or low middle income country, it also would be a commitment from the other side to basically help us to do upfront investments as well. Yeah, but we're very open and we're in current discussions with our key customers. Yeah.
Thank you very much. I think that we can come to a close. I wonder whether each of you could give one sentence about take-home message.
I can go ahead.
Yeah.
Yeah, I think again, thank you for helping organize this. I think what again we've sort of seen and was clearly really important is collaboration among the industry to really help industrialize and bring these therapies to more patients. So we need to continue to do that and hopefully can make progress on standards where it makes sense to make this easier for all of us to manage.
Thomas?
Yeah. If I may follow. So first of all, thank you for allowing me here to be present in that panel. DHL is very committed to the life science sector. in different formats, be it now with the vaccines rollout, but also in co-investing together with customers and sponsors. And we have obviously an eye on all of these trends like cell and gene therapy as well. And yeah, supporting basically our customers to be successful here in this area as well. So thank you.
Thank you. Robin?
I think just to reiterate, standardization of approach from industry to us would be enormously helpful. I mean, our future is just more and more complicated, onboarding, stem cell lab, all of this stuff. It would just be really great if there was a sort of a uniform approach from different parts of industry.
Ruben?
Yeah, I mean, again, thank you for the opportunity of speaking today. And I want to just kind of make a point about the collaboration that does exist in the cell therapy world between academics, the industry, government bodies who are approving these therapies, and that's a fantastic thing to have. And I think there needs to be more of that going into the future. I think just as a final point when we're developing all these cell therapies is just to ultimately still make sure that we're putting the patient at the center of all of this. So, there may be solutions that might work well for a clinician or for industry, but not necessarily for patients. And I think we mustn't forget that.
Absolutely. Rick, shall I hand it back to you to close? Thank you all.
Thank you very much, everyone. That was a really fascinating presentation and discussion. Hopefully everyone has taken away something. Apologies for any questions that we probably might not have got around to. However, what I will suggest is if we have, or rather if there were any questions that you'd like to have any follow-up on, please do send them through to myself or Katerina. We will drop our emails in the chat. And we can then follow up with the speakers in due course. What we will then look to do with the recording is we will send a link around once we have uploaded it to the ATM platform, and we will go from there on, hopefully within the next week. So many thanks to everyone for joining, and take care.